let's quickly look at the theory of the 2023 past question to see what we have got as we prepare for the exam. So the first question here says, states the cell theory. This is the first question they asked them. The next one says, name two scientists that contributed to the development of the cell theory. So you see, if you have watched our review video, you'll see that these questions were treated in details. So do well to go back and see those videos for yourself if you've not seen them. If you're new, thank you for joining us. If you've not subscribed, kindly help us to click on the subscription button so that your friends and your classmates also can see these videos as we prepare for our exams. The next one is state 5 characteristics of an entomophilous flower. So basically this word was just to confuse you. Entomology is the study of insects. So entomophilous is basically insect pollinated flower, right? And then state the organ of vegetative propagation in ginger, banana, sweet potato. This is the question they gave them. And then the next one say describe the process of digestion of bread from the mouth to the anus. So let's see the answers before we come back to continue from there. Looking at this very closely, we've done this over and over again. I'm not going to waste time here. The cell is the basic structural and functional unit of life. All living things are made up of cells. New cells arrive from pre-existing cells by cell division. And then the scientists that contributed were your Matthias Kellerden, your Theodor Schwann, your Rudolf Virchow, and your Robert Hooke. Okay, I didn't include that, so let me just write that there. Robert Hooke. Okay, so you need to take note of the spellings. Characteristics of entomophilous flower: insect pollinated flower, large petals, brightly colored petals, sweet scent, produces nectar. The pollen grains are large, rough, and sticky, and the stigma is usually inside the flower. Okay, so please take note of that. The next one here is the vegetative propagation. In ginger, you have the rhizome. Banana uses the sucker, and sweet potato uses the tuberous roots. Okay, so take note of that. And then finally, before we continue, the process of digestion of bread from mouth to anus. The bread is chewed in the mouth, mixed with saliva. The amylase, salivary amylase we talked about in the objective of this particular review, changes the starch to maltose. The food passes from the esophagus to the stomach by peristalsis. In the stomach, the food is churned. There's something called churning in the stomach and mixed with gastric juice. In the duodenum, or the first part of the small intestine, the pancreatic amylase continues the starch digestion. Intestinal enzymes convert the maltose to the glucose. Glucose is then absorbed in the villi, and then undigested food passes out through the large intestine where water is absorbed and expelled via the feces. So you want to take note of this. This is the process of complete digestion of anything carbohydrates from the mouth to the anus. So you want to write that down. That's very, very important. Or pause and try to read to understand for yourself. Okay, moving on to the next question. The next question says, using arrow diagrams, rearrange the following in order of their increasing complexity. So they gave us tilapia, hydra, snake, amoeba, and toad. So the next one now says 3 features of Tridax procumbens, fruit that makes it adapted for wind dispersal. Okay, remember we said Tridax is dispersed by wind. It was part of the objective question. So they were just asking you answers in the objective and the theory. This was a very simple year, and yours is going to be much more simpler actually. Describe an experiment. You must know this because they asked 2 of this particular year, so you must go and learn how to describe simple experiments to show that describe an experiment to show that the shoot of plant is positively phototrophic under the following heading. So you must put your aim, your apparatus, your method and procedure, observation, and then your conclusion. You must talk about those parts to get your marks. The marks will be shared along that. And then here they asked us to, in a tabular form, 2 differences between aerobic and anaerobic respiration. All these things were captured in our review video. So if you've not seen the video, try and see it before you go into the exam hall. That will be one of the best things you would have done for yourself. Okay, so arranging them now in the order of increasing complexity, you have amoeba first. Why? It's unicellular, right? You now have your hydra. Okay, you now have your tilapia, you have your toad, and then you have your snake. So that's basically the hierarchy of arrangement of these organisms. Okay, hydra operates at the tissue level. This is the cellular level, but in the classes of chordata, you actually have your Pisces before you have your amphibians, before you have your reptiles, then you have your apes, and then you have your mammals. in the increasing order of complexity. So take note of that. 3 features of tridactyl cucumbens that helps them for wind dispersal. Basically, they have feathery hairs called the papals. They have, their fruit is very light in weight, and then they are small and very dry, so they can be carried easily in the wind. Now they asked us to talk about the experiment to show that the shoot is what positively phototropic. So the aim is to show that the shoot grows towards light. That's what it means to be positively phototropic, meaning that if it is positive, it's going towards light. If it's negative, it's going away from light. So the apparatus now, we mentioned a few things: the healthy potted plant, cardboard with a hole of light on one side. Then the method and procedure: you place the box, place the potted plant inside the box, allow light to enter only through the hole, leave the setup for days, and then observe the direction of the words of the growth of the shoot. So that will now tell you if it is growing towards the light or if it is not. The observation, what you would see at the end of the leaving it for some days, the shoot will begin to bend towards the opening where you have the light. So that's your conclusion will now be that what the shoot responds by growing towards the light, therefore it is positively what phototropic. From your experiment, you can now make your conclusion. And then the differences between aerobic and anaerobic respiration: this one requires oxygen, this one does not require oxygen, this one provides much energy, this one provides very little energy. Okay, and so that's that. So I think we have covered the questions there. Let's move to the next question. We are trying to make this very, very brief so that you can just be very fine and review for yourself. The next one list 2 examples of freshwater habitats. In a tabular form, state 2 differences between complete and incomplete metamorphosis. 4 marks. You must always make sure you give them points that are in line with the number of points they've given you. Another experiment is coming here, so pay attention. This is why I had to go and do this review, because I felt I should just tell you guys this thing as you prepare for the exam. Now, this one is to show that chlorophyll is necessary for what? For photosynthesis to take place. The same thing: aim, apparatus, method and procedure, observation and conclusion. This question, let's see how to tackle this. Now, the next one: state 4 harmful practices that deplete soil fertility and enumerates 2 effects of noise pollution. NECO love a lot of things around pollution, conservation, natural habitats and all of that. So please read up that. And then maybe we should just look at the answers before we come back here. So let's look at number 3 answers. Number 3, 2 examples of freshwater habitats. Basically, you have your pond, you have your river. The difference between complete and incomplete metamorphosis. So complete has 4 stages. It has the egg, has the larva, has the pupa, and then finally ends with the word adult. Either you call it the adult or you call it the imago. It's the same thing actually. That's the last stage. But the incomplete has 3 stages, and the 3 stages are the egg, you have the egg, you have the nymph, NYMPH, and then you have the word adult. So we see now that the second difference is that the young differs greatly from the adults, the larva. But in this case, the nymph resembles the adult. It's just that the nymph does not have a reproductive structure, and if it's for a winged insect, it doesn't have wings. Exactly. So please pay attention to that now. So that's how to answer that question. Pay attention to that. The next one, the experiment to show that chlorophyll is needed for photosynthesis. Remember your aim. So your aim will be to show that chlorophyll is necessary for what? For photosynthesis to take place. What will you use? You use your variegated leaf, your beaker. A variegated leaf is a leaf that has a green and white patches. So it means at the end of this experiment, if you are testing the leaf having green and white patches for starch, the part that are green will be positive for starch because that one has chlorophyll. But the part that are white obviously will be negative. There is no chlorophyll there. So starch cannot be generated in those areas. So you use ethanol, iodine solution, forceps, Bunsen burner, and then your hot water bath. So basically your procedure: expose your discharged variegated leaf to sunlight for a few hours. Discharged means that it did not contain starch, but exposing it for a few hours will now make them to get starch. Plug the variegated leaf, boil the leaf in water, boil in ethanol to remove the alcohol. Normally when we are removing the ethanol, we use when we're removing the chlorophyll, we use ethanol. We boil the leaf in ethanol, rinse in warm water, add iodine solution. Okay, so you see now that our observations, like I earlier mentioned to you, the green parts turn blue-black, whereas the non-green parts will remain brown or yellow. So the non-green part or the white part basically will retain the color of the what of the ethanol of the iodine solution, which is blue black. And then what will be your conclusion? Only the green parts containing chlorophyll can manufacture what? Starch. Therefore, chlorophyll is necessary for what? For photosynthesis. Very simple exam. Simple exam. Okay, so don't even fret, don't even bother yourself too much. Just know that what you have studied will come and is going to come out very well. Now, state 3 principles to be observed in crop rotation system. You see, tilting towards a bit of agric, and then list 5 adaptive features of xerophytes. Xerophytes, I told you in the objective test that they are what? Dry plants that do well in dry areas, like your cactus. Enumerate 3 evidence of convergent evolution to show that birds might have descended from reptilian ancestors. So convergent evolution, evolution that tilts, that comes towards a point. Okay, so that is convergent evolution. 2 different species coming close to become increasingly similar by the day. That's convergent evolution. The next one, list 5 functions of the liver, and then give 4 methods of conserving forest resources. So please pay attention to these topics. These topics are I don't just know why NECO loves this topic a lot, but they ask questions a lot around conservation, pollution, and natural habitats basically. So number 3, harmful practices that deplete the soil fertility. You have your bush burning, overgrazing, deforestation, and continuous cropping. So these are captured there. And then 2 effects of noise pollution, basically hearing impairments or deafness, and then you have stress or loss of concentration. Just write these things down. These ones are basic things you can write from your head actually. And then 3 principles of crop rotation. Crop rotation is when you so let's just look at some of the principles to answer the question. Number 1, include legumes in your crop rotation. Why? Remember the nitrogen cycle. We talked about it in the earlier video we reviewed. So go and look at that video. The nitrogen cycle, legumes are plants that help to fix nitrate in the soil. So when you want a soil, a particular vegetation to be fertile, or a soil to be fertile rather, you would want to plant legumes. So planting legumes will increase the nitrate concentration, okay, and that's because of nitrogen fixation by a rhizobium leguminosarium. You should know that when you study the nitrogen cycle. They do not, so include legumes in your rotation. Do not grow crops of the same family consecutively, okay, and then alternate deeply rooted crops with shallow rooted crops, so that the deep rooted ones can get the nutrients from the lower parts of the soil, whereas the shallow rooted ones will get from the upper parts of the water of the soil. That's very important. So adaptive features of xerophytes, you have the waxy cortical. Remember in the objective there's a question they asked us that plants in tropical rainforest, we see they don't really have a thick stem because they don't have much needs of conserving water. Reduce leaves and spine, that's because they want to reduce water loss by transpiration. Sunken stomata, and they also have fleshy stems for water storage. So these are all characteristics or adaptive features of xerophytes, plants that are very that do well in water loving areas. So that's for that question. So what else are we looking at? Okay, let's look at the last question. A pure breeding dominant white colored rabbit. Please pay attention to this because this is the first genetic question I'm actually handling with you, and this is one of the reasons why I had to come to this past question review when I saw this genetic question. So I would want that you pay attention. You see, it has a whole lot of 8 marks, so you need to pay attention and know what you're doing. A pure breeding dominant white colored rabbit, so you have capital W, was crossed with a pure breeding recessive black colored rabbit, small w. By means of crossing, show the genotypic and phenotypic ratios of the F1 and F2 generations. So we finish the answers in looking at the other one. So the 3 evidences, we'll go back to the pure breeding stuff, but let's just finish this. 3 pieces of evidence that birds descended from reptiles. Basically, you have that both of them they lay shelled eggs, both of them they have scales, and then both have a cloaca. Cloaca is also found in both birds and reptiles. Then 5 functions of the liver, of course, production of bile, storage of glycogen, detoxification of harmful substances, deamination of excess amino acid, and storage of vitamins and iron. Okay, so please pay attention to that. And the forest conservation, is basically by afforestation, planting of trees, reforestation, replanting of trees, controlled logging, and then establishment of forest reserves, areas where you don't go to cultivate at any means. So I needed to say that before we move on to the crossing, the genetic cross we had. So this is now the genetic cross. So they told us earlier that this man, okay, pure breeding, capital W, was crossed with small w. So normally when you do this now, this is a parent genotype, okay, this is a parent genotype. Now, but you have to separate them into gametes. So doing that well, you're going to put this now, put your capital W inside, another one, put another capital W inside. You are now crossing it with a small w. So these small ones now I'm putting them in small cycles are called the gametes. So these are my gametes. And this is the first law of segregation of genes. The alleles appearing together, they separate into genes during meiosis. So what now happens is that it is these ones that you cross. So crossing this now, you bring in this and bring in this. If you are having this together, you are having capital W and small w. You are now bringing this one and bringing this again. Doing this, you have another capital W and small w. Bringing this and bringing this again, you're going to have capital W and small w. And then here again, you have another capital W and small w. So all of them will be what? Capital W and small w in this F1 generation. This is the first generation, so all of them. So it means that all the offspring for the first generation will be what? Capital W and small w. That's why I told you that for the F1 generation, these were the gametes I listed. For the F1 generation, all of them, the genotypic ratio is going to be 100% because you have 4. So 4 over 4 times 100 is 100%. Okay, so that's why I said here that you have 100% capital W, small w. Okay, and the phenotypic ratio, obviously they will carry the dominant character, which is the character of this dominant parent. So all of them will be white, 100% white. Remember, phenotype is what you see. What you see is phenotype, the outward expression of a gene, but genotype is actually the genetic composition of the organism. Now let me stress the F2 generation. So for the F2 generation, I'm going to pick 2 members of F1 generation and cross them. So I remember I'm going to have this capital W, small w, cross with another capital W and small w, because I'm trying to pick from F1. These are members of the F1 generation. I'm trying to cross to get the F2 generation. So the same thing we did earlier to separate them into the gametes basically. So I'm going to have my capital W here, and then I'm going to have my small w here, trying to make it small so you can really see. Another capital W here, and then this is going to be another small one. So this, let's now see the offspring you have for the F2 generation. So I'm going to bring this and I'm going to bring this. So that's going to be a capital W, follow me. And then this coming again with this. Now that's going to be a capital W and a small w. I'm trying to differentiate the letters. And then here and this, now I'm going to have another capital W and a small w. And then finally this guy comes to meet with this guy, and you have your small w. So you now see that it's different. In this ratio, I'm now going to have 1:2. Where is it 2? 1 and 2. These 2 were capital and small, okay, and then it's to 1. This one is small. So I'm going to have capital small and small. So that's why in the F2 generation, I'm going to have my genotypic ratio to be 1 capital W, 1 capital W, 2 capital W, 1 small w, and 1 small w. So in this case now, phenotypic ratio is what you would see. This one is going to be white. These 2 also are going to be white because that's what you're seeing, because the dominant gene is present. But this one that is small w, for the first time is going to be black. Okay, so that's why I'm going to have 3:1 as my phenotypic ratio. That's what I carefully listed here for you. I just tried to use the genetic cross to explain these concepts to you. So please go ahead and learn about this. There are so many things you can learn about this: probability in genetics, test cross, back cross, dihybrid cross, and the rest of them. Don't just trivialize this. Try to see how you can learn and know these things for yourself. They'll be very fine. And we don't want to make this video very long. I think that's the last. Let me see if there's another question here. State 2 functions of the calyx in plants. Name 5 animal hormones, and then enumerate 2 ways in which the principle of heredity has contributed to the field of medicine, and then give 3 structural adaptations of leaves, for photosynthesis. So that's the last. Let's just run through the last page for the answers. So here, functions of the calyx. Calyx is a collection of sepals. So it protects the flower in the bud stage, supports the petals when the flower is open. And then let's move on to the last one for today. 5 animal hormones you are having: insulin, adrenaline, thyroxine, estrogen, and progesterone. You need to know what they do. You need to know where they are secreted. I need to know what they do. Go and check them out. They are called endocrine hormones, basically. They are secreted by the endocrine glands. And then 2 contributions of heredity to medicine: detection and prevention of hereditary diseases like your HIV and the rest of them that the research is still ongoing, your hepatitis, okay. The knowledge of heredity has helped in that regard. And the next one, blood grouping for safe blood transfusions. Okay, so that's the other ones, but these are the 2 I just listed. And 3 structural adaptations of leaves for photosynthesis: broad lamina provides a large surface area, numerous chloroplasts containing chlorophyll, and then the thin leaf prevents or allows rapid diffusion of gases and penetration of light. So that's how far we can go on the review. At least I tried to show you some basic answers. So sincerely, I wish you all the best. I want you to do well, and that's why we are doing all of this. We had to put out like 3 videos in a space of less than 24 hours just to make sure that you prepare very well for this exam. So it is my desire that you do well, and then you would help us to share this video, help us to like, comment, and subscribe if you've not subscribed, so that this video can reach as many persons as possible. We wish you all the best in your exams, and I hope that you would come back to share the testimony of how our little research and study has helped to increase your chances of doing very well in the exams. Thank you very much. My name is Emekudo, and I'm going to see you in the next one. Bye for now.